And under family Pasteurillaceae, we have genus Pasteurella, Actinobacillus, and Haemophilus. And we talked about diseases that are important under family Pasteurillaceae. The important species is your Moltocida. I will highlight some of the important parts of the lecture. So last time we talked about the virulence factors, the importance of the capsule to your Pasteurella multocida. If it doesn't have the capsule, then it will be less pathogenic or it will cause less mortality. In this case, 0% mortality if we remove or delete the capsule. And then we talked about some of the diseases that are important caused by your Pasteurella multocida. So in cattle, there's hemorrhagic septicemia. Then we have fibrinous pneumonia and atrophic rhinitis in swine and some other species, fowl cholera in birds. No? Then we looked at some of the pathology that is caused by Pasteurella, and then the lab diagnosis, treatment, and control. Then we discussed Ornithobacterium as well. For Ornithobacterium, there is only one species, that's Rhinotracheale. It is a fairly recently discovered disease or species. The pathology is fibrinous air saculitis pneumonia. And we looked at some of the gross and microscopic pathology that is seen in this bacteria. Then last one is the Bordetella. We emphasized two species, but one is in humans, the pertussis. This one affects young children. It's important for it's important in young children, no? Then there is a species called Bronchiceptica that is important in animals, no? So yung two important diseases would be the atrophic rhinitis in pigs, and in dogs we have canine infectious tracheobronchitis, or what is known as kennel cough. We mentioned that this bacteria is a strict aerobe, no? So mostly it affects the upper lung areas. It does not uh it it requires oxygen, so it does not go into the deeper parts of the lung where there is less oxygen, no? Unless there is a viral infection, a co-infection with the virus, then the pathology can look worse, no, or it can worsen. Okay, so that's Bordetella. Now we'll continue with the rest of the topics for this week. We have your Moraxella and Reimerella, and then we will see if we can finish Actinobacillus, Streptobacillus, and Francisella. But I think we'll save some topics for Wednesday so that we can still use up our Wednesday class. Let me look at the calendar as well. So yeah, Wednesday na yung last class ko with you guys. Next week is apparently reading break, and then the rest of the sem will be handled by Dr. Reyes and Dr. Colorado. Okay, so let's proceed to Moraxella and Reimerella. Are there any questions so far with the three ones that we reviewed today? Okay. Quick question. Um, is it okay if I give you a online quiz this week or masyadong maraming ginagawa? Um, let's admit everybody first. Okay lang. Okay, good. Thank you. Ox lang, no? Kaya nyo yan. Okay. And then also, have you guys accessed the lecture? Have you um the lectures last week, were you able to access them? Okay. Uh, can you let me know if any of you guys had accessed the materials for last week? Or siguro hindi tayo nag-lecture. Ay, nag-Zoom lang tayo. I think yung 4 to 5 class pala yung recording lang yung na-provide ko. It's okay. You don't need to answer that question. Okay. Sige. So let's go to Moraxella. So for Moraxella, this bacteria is associated with the eye. No, this is the appearance of the Moraxella again. This okay. Para ano, may interaction. What is this stain? Is it gram positive or gram negative? Okay, thank you, Joanna. Correct. It is gram negative, and the appearance of the bacteria is Coccobacillary, no? Okay, so this bacteria, Moraxella, was named after a Swiss ophthalmologist named Victor Morax. So he was the one who discovered this bacteria. It's a gram-negative aerobic non-motile bacteria, and it has the coccoid rod or coccobacillary morphology. So they appear singly, in pairs, or in chains. And there are two major species. No? This is what it looks like um, up close. This in particular is your bove oculi. The bacteria produces a hemolysin, and it also has type 4 villi. This type 4 villi is responsible for the adherence of the bacteria to corneal epithelial cells. So if you remember the histology of the eye, the cornea is covered by epithelial cells, and this is where your bacteria will adhere to or bind to. And the type 4 villi is very similar to other bacteria such as your Pseudomonas aeruginosa, Neisseria, Dicolobacter, Pasteurella, and Vibrio cholera. There are other enzymes active, which are able to break down the lipids, peptides, and carbohydrates that are present in the eye. The bacteria can be isolated in the lab. It's an aerobe, so it does not require any special conditions for growth, and it grows best at 35 degrees Celsius. Within 48 hours, the growth of colonies can be seen. The nitrates and urea are not utilized, but it digests proteins, and it is susceptible to commonly used antibiotics. So this is a bacterial infection of the eye that is treatable. No? In culture, the colonies will lack the pili because it doesn't need it for attachment. And there are some non-hemolytic variants that are non-pathogenic. So here's what the Moraxella looks like under or in on the plates. No, so they have small, dry, flat, friable colonies, and they appear to corrode the agar. The disease that it causes is infectious bovine keratoconjunctivitis, or what is known as IBK. Another term for it is pink eye, mostly because the eye becomes inflamed and it causes redness of the eye. The two species that are associated with this disease is your bovis, Moraxella bovis, and Moraxella oculi. Sorry, this should be oculi, oculi. Okay, and this is a recently discovered species. It was only in 2007 where when it was characterized. So the two main virulence factors would be the pili and the hemolysin, which allows the attachment of the bacteria to the conjunctival epithelium. The bacteria also shows some leukotoxicity for macrophages. However, if you think about it, um, the eye is also known as immune privileged. No, so I want you to look up this term. Um, I'll put it in the chat. So when we say immune privilege, um, or it's an immune privileged site, what do you think it means? No, from the two words, no, immune privileged. Um, what is the meaning of this? Can anyone in the chat try to answer that question? When we say the eye is an immune privileged site, what does it mean? Have you ever heard of the term immunoprivileged or immune privileged? They are protected from immune response. Okay, so it is not included in the body's normal immune response. Yes, no. So it means that there is a reduced number of cells or immune cells that are present there. But it doesn't mean that it doesn't get inflamed. No, it can still be inflamed or it can still have an immune response, but it is significantly reduced, mostly because the body is trying to protect that organ, no, or that tissue. So the eye is essential to the biological being or to a creature or to humans, diba? Or to an animal. It's an essential part of the the body uh, to function. So the body tries to protect it by reducing any form of immune response that may be present so that um it doesn't get affected, no, when there's let's say systemic infection, or at least it tries to reduce that. There are other parts of the body that are immune privileged. No, it's not just the eye, but The brain, for example, it's immune privileged, and the reproductive organs, for example, the testis is immune privileged. No? So there's less inflammatory cells that are present there. No? And so um, but the bacteria, no, this bacteria, it still it still has a defense mechanism against the immune cells, such as macrophages. No? And then there are other enzymes. We mentioned this kanina, the lipopolysaccharides, collagenase, and hyaluronidase contribute to its pathogenicity. For the pathogenesis of the bacteria, first, it will attack the eyes which have been irritated. No, so if there are any factors that would cause irritation to the eye, then this would worsen the eye infection. These factors would include UV irradiation, flies, dust, woody pasture plants, and other viruses or bacteria. Even your parasites, they can also worsen the infection of Moraxella bovis. So this occurs mostly in animals that are in pasture, and they could be exposed to the environment. The viruses would be BHV. So IBR is caused by bovine herpes virus 1 or BHV. And IBR means infectious bovine rhinotracheitis. And then adenovirus that can also co-infect with Moraxella. Mycoplasma and Listeria can co-infect this bacteria as well. And then nematodes, the bovine eyeworm is your Tillassia. This is an eyeworm. So what happens is that the bacteria will invade the conjunctiva and the cornea and will cause edema and a neutrophilic inflammatory response. The animal will develop mild epiphora or tear formation. Then eventually it will spread on the corneal epithelium, causing clouding of the cornea, severe edema, opacity of the cornea. Eventually, because of the inflammation as well, there will be vascularization, ulceration because of the enzymes, breaking down the tissue. And then it could lead to uveal prolapse, and the entire eye can become inflamed, causing what is known as panophthalmitis. But the bacteria is known to be self-limiting, so it will heal eventually. So the ulcer will heal. There will be central scarring, and eventually, but during this process, there may be vision impairment. Mostly the negative effect of this is that the animal will have difficulty in foraging. So let's look at the clinical stages of IBK. This is stage 1. So there will be epiphora, sensitivity to light, corneal ulceration. In here, this is seen as the white spot right here. And so this is the corneal ulcer, and then the some parts of the cornea are cloudy. So this one is an inflamed cornea. Antibiotics will be effective. So we can give long-acting tetracycline injections. And then we have stage 2, wherein the iris can still be visible, but vascularization is evident. And so here we see the redness of the eye because of the vascularization. Still, we can provide antibiotics to this, but you may have to perform a conjunctival injection. So for the bulbar conjunctival injection, we'll look at how it is performed later. And then you have stage 3, wherein the eye will have severe ulceration. The entire eye could be affected. So here we see panophthalmitis, and there is some yellow coloration right there, showing the pus formation, no, fibrin and pus filling the eye. In this case, we can still provide antibiotic, bulbar conjunctival injection, and an eye patch using the third eyelid. And in worst cases, there will be corneal, the the ulcer will rupture. So there will be corneal rupture, and eventually the uvea will prolapse out of the cell, and will this will lead to partial or complete blindness of the animal. So that's stage 4. This is how a bulbar conjunctival injection is performed. So it's dun sa lateral uh, side of the eye, no? And this provides local injection of the antibiotic to the eye. Okay, so let's try to practice with the scoring, no? So kanina we said there's 4 stages, 1, 2, 3, 4. Based on that discussion, can you tell me what do you think is the stage that this eye is in? Is it stage 1, 2, 3, 4? What do you think? Okay, so some said it's stage 4 or stage 3. I would say this one is stage 3, mostly because the uvea has not yet prolapsed. And so there is vascularization, there is cordial ulcer, but there's no uveal prolapse yet. No? So we would say this one is stage 3. So based on the stage, then you would know what to do, right? So um, ano ba yung recommendation? So, for stage 3, we said can give antibiotic, we can provide the injection, and then we can also cover the eye with the third eyelid. Okay, what about this one? What stage do you think this is? Stage 1, 2, 3, 4. Okay. Okay. So stage 4 kasi there's the entire eye is affected. So there's panophthalmitis, probably blindness na, no, in this case. No? Okay. So that's Moraxella bovis, or infectious bovine keratoconjunctivitis. Okay. Any questions with this topic? Claire is in the waiting room. Okay. Oh no. Let's proceed to Reimerella. So I think we'll cover until Actinobacillus today. So we have enough time to finish all of the materials for this week. Okay. So the next genus is your Reimerella. So what you need to remember about Reimerella is this affects ducks. You will see the gram stain here. Can you tell me gram positive or gram negative? Gram negative. Okay, good. That's correct. And this bacteria, it was formerly classified into various genera until it was able to or it until it was classified into its own genus. So yung former classification niya was Moraxella, Pasturella, and Pfeifferella. But now it has its own genus, which is Reimerella. There is only one species you need to remember, which is Anati pestifer. And this is seen in ducks, no? So this is the causative agent of new duck disease. Mostly young ducks and turkeys are affected. So yung mga waterfowl, chicken, and pheasants, they can be affected. We want to be aware of this bacteria mostly because devastating siya to duck industries. And alam naman natin dito sa Philippines, we have a duck industry kasi kumakain tayo ng balot. So we want to be aware of this Reimerella anatipestifer. So this infects the respiratory route or through the percutaneous, such as in the webbing of the feet and ducts. So this is what it looks like. So small rods, and they can also appear filamentous, no? Just like the one. There's a gram-negative picture, like this one. Other names would be duck septicemia, goose influenza, infectious serositis, polyserositis, and rhinorrheliosis. So those are the other names of new duck disease. It's a non-motile, encapsulated, and occasionally bipolar coccobacillus or thin rods. This bacteria is microaerophilic, and so it requires the increased CO2. Then you have to provide blood agar or chocolate agar. Some are non-hemolytic, some are hemolytic. They have transparent colonies and may exceed 1 millimeter after 24 hours incubation. For the pathogenesis of the bacteria, there is sudden mortality, and there can be two major signs. No, there are the respiratory signs, and then you have the neurological signs. However, you can see the diarrhea as well. So yung GI, pwede rin siyang affected. So for the respiratory signs, this includes sneezing, coughing, nasal, and eye discharge. And then for the neurological signs, there's head tremors, torticollis, and ataxia. Um, okay, somebody needs to leave early. Okay, you can go. The recording will be provided. And then head tremors, torticollis, ataxia, yun yung sa neurological signs. There is so the disease is mostly acute. So there's an acute phase. There is fibrinous polyserositis, and so multiple organs can be affected. Exudates appear to be mononuclear, and fibroblasts and giant cells can be seen in protracted cases or in prolonged cases. Some people still in the waiting room. Okay. 1 to 2 several weeks duration. So the disease can last this long, and the mortality can reach up to 75%. So this can be economically devastating in case it's present, no, in some cases. And here's um a a duck with some of the signs. So we can see here that the feathers, no, they don't they appear to be ruffled. Um it's also sitting. So normally, 'di ba, hindi naman so normally yung ducks when they're on land, they're usually standing, no? So in this case, it's probably suffering from ataxia. And then we have the diarrhea. So a lot can be said about this photo. Then we have the polycerositis. So in the abdominal cavity, we have a number of organs affected, and then you have the fibrinous exudates. No, so if you notice, a lot of the diseases they will have the fibrinous exudates covering the organs. No, in this case, the liver is most prominent, and then we have the fibrinous exudation. Then you have the joints; they are swollen, so this will cause the ataxia or the ataxia. And then recovered birds are resistant to re-exposure. There is serotype specific immunity, and then there's 12 serotypes known. So when we say, does anyone know what is the meaning of serotype specific immunity? Can anyone tell me what that means? Serotype specific immunity. Adaptive immunity to a specific serotype of bacteria. Okay, so it means that your body is able to develop a um, response that is able to target that particular serotype, right? And it is only specific to that serotype, meaning that if there's another serotype that infects you, do you think that you are immune to that or not? Let's say you were infected by serotype A, and then you got infected, and then you developed a specific immunity, a serotype specific immunity to serotype A, right? And then you got infected with serotype B. Do you think you'll be immune to serotype B? Yes or no? Okay. Mostly everyone got, or most of the people who answered got it correctly, or all of the people who answered got it correctly. So no, no. So that means that your body will not have um, a specific response to serotype B yet. No. So, hindi pa. Pero for serotype A, meron ka ng immunity. Now, I need you to remember that kasi there are many types of bacteria that have this uh, type of immune response. No? So, yung leptospirosis, for example, the immunity for leptospirosis is serotype specific. No? And so, pag nagbakuna ka for a particular serotype, kung naapektuhan ka ng ibang serotype, hindi ka immune doon sa ibang serotype, no? Nagets ba 'yun? Klaro ba 'yun? Sabihin, yes, okay. Then, we have bacterins, may induce immunity 'yun. So, there are some bacterins that may induce immunity but, hindi siya widely distributed, no? And it's not normal practice for us to vaccinate against this particular bacteria. Siguro pagka nagkaroon ng incursion, then maybe we need it. For lab diagnosis, the organisms can be demonstrated from organs, fluids. Then you can perform gram stain or in isolation of the bacteria or even immunofluorescence. So si Reimerella, it is known to produce biofilms. So we mentioned biofilms in other bacteria. For example, in Pseudomonas, no, si Pseudomonas nagpo-produce din siya ng biofilms. And this is how biofilms are formed. So, remember the capsule, this is a carbohydrate-rich biofilm. No? And first, the bacteria will attach to surface, and then it will form the biofilm. And what it looks like is a community of bacteria. So this is slimy, no? So yung ano nito, itong biofilm na to, slimy siya. And yun yung nagpa-protect dun sa bacteria so that it can persist and it can move to another location. So when there is detachment, so that's the third stage, this releases the bacteria and then it allows the bacteria to move to another location. So for the treatment and control, antibiotics, so you have sulfonamides, Pen-G, erythromycin and novobiocin. So in general, these are antibiotics um, that can affect your gram-negative bacteria. Then there's vaccination with killed or live attenuated vaccines. But again, here in the Philippines, there's no known outbreak of Reimerella, pero I think we need to be aware. Then rigid sanitation and depopulation. Okay. Kasi may resistance siya eh. So, dahil sa biofilm. Okay, any questions with regards to Reimerella? Okay. Then, I think we have enough time to cover Actinobacillus. So, for genus Actinobacillus, there's a number of species that you need to remember. Okay. So, kita nyo naman, gram-negative siya. And for the morphology, Coccobacillary. So, there's cocci and rods. Here we can see the rod shape. Some of the characteristics would be production of surface slime. So they have sticky colonies on agar. This is seen in Suis, Equili, and Ligneraceae. Then there is a species called Pleuropneumoniae, which was originally Haemophilus, which is encapsulated. This is a bacteria that's very phenotypically similar to Pasteurella. So, halos pareho siya sa Pasteurella. And it is a frequent commensal or parasite of the mucous membranes. Most actinobacilloses are endogenous infections. So if you remember yung discussion natin about Pasteurella and Haemophilus, no, um, the infection is endogenous, meaning that the bacteria is already present in the body of the animal, right? Except in neonates, they can infect the neonates, and then foals acquire Actinobacillus equili via the umbilicus. So very similar din siya dun sa Haemophilus in that uh, um, um it will use the blood agar or chocolate agar for growth. So they have grayish white colonies on chocolate agar, produces a thin zone of hemolysis on blood agar, and it grows at 20 to 42 degrees. It can grow on maconchi but dies within a week. Just like your Haemophilus, this will demonstrate satellitism. That means it requires a helper bacteria such as Staph aureus, which will release the factor 5 and factor 10, also known as NAD or NADP and hemin, respectively. And this is the Staph aureus, no? So yung white. And then the Actinobacillus grows within this quadrant wherein these two factors are supposedly released. So mostly nandito sila. And here as well. So the chocolate agar, once again, if you heat the blood agar, it will become chocolate agar. That's 80 degrees Celsius. It will release the two factors required by this bacteria. Some uh, medical cellular products of medical interest, the RTX gene or the repeats and toxin gene. There are three major hemolysins, no, that are produced. EPX 1, 2, and 3, and they kill the macrophages and neutrophils. Just like other bacteria, it has the pili for adherence. In this case, it goes into the respiratory system. So it will attach to the alveolar epithelia and cilia of terminal bronchioles, especially for APP. So when we say APP, that's Actinobacillus pleuropneumoniae. So in the field, you might hear people saying in the swine industry, APP. So this is what they're referring to. Then you have LPS, since it's a gram-negative bacteria, the OMPs, and the Actinobacillus ferric uptake. So this is for iron transport. So if you recall, your gram-negative bacteria, they take up iron and they need a system for that. And in this case, it's the Actinobacillus ferric uptake. So do you recall what's the term for this? What's the term for the iron uptake system? Starts with the letter S. Siderophores, correct. So this is the siderophore for the Actinobacillus. And then you have antiphagocytic capsule and resistance genes. No? So they will carry the R plasmids or resistance plasmids for these bacteria. So it's very common for them to have the resistance. At high concentrations, RTX toxins will damage the macrophage and the neutrophils. And at low concentrations, the phagocytic cell activity is increased, which damages the neighboring cells and triggers inflammation. The LPS will induce an inflammatory response, so it will trigger the release of interleukin 1 and TNF. And infection spreads via the lymphatics. So remember, some bacteria will spread via the blood. This one will go through the lymphatic system, and it will produce these granulomatous lesions, especially in chronic cases. So there's a number of species you need to remember based on the host affected. So your Actinobacillus lignaresi, it causes wooden tongue in bovines. It may affect dogs and rarely horses. This causes a chronic granuloma. Then you have horses. It will cause navel ill. The species is Equili. So some symptoms would be enteritis, suppurative multifocal nephritis, tendinogenitis, arthritis, and enteritis. Actinobacillus suis, this will cause various inflammatory processes. Yes, for those who need to leave, you may go. We're almost done with this slide, so you can just look up the video if you need to come back to it. Okay. And then lastly is your pleuropneumoniae. This is the APP. This is the most important one in swine industry kasi it will cause the granulomatous pneumonia. Okay. Now let's look at some lesions of APP. This is a consolidated lung lesion of a 3-month-old pig infected with APP. So you may see the bacteria can affect the deeper lung tissues, no? And it will cause these granulomatous lesions. So when you say consolidation, what do you think is the meaning of that? Consolidated. Lung consolidation. What is the meaning of that? So napansin niyo may mga patterns yung lungs, 'di ba? Depende dun sa bacteria na nakaka-affect sa kanya. So, what do you think is lung consolidation? Anyone in the chat? The lung becomes dense. Uh, that's correct. Or hardened, no? So the normal consistency of the lung is sponge-like, 'di ba? Kasi may air siya sa loob, no? But when it becomes consolidated, then it becomes hardened, meaning that it's filled with inflammatory cells. It's inflamed. It's thickened, no, and it has exudates, no. So, yun yung lung consolidation. No? So basically, pneumonia siya. The lung is inflamed, and then there are areas that are hardened, no. So there's lung consolidation. And here's some lesions associated with APP in the or microscopically, no? So here we see some microgranulomas, no? And these are larger granulomas that are seen in the lymph nodes. So these are enlarged pulmonary, hilar, and mediastinal lymph nodes of the pig. These are produced by the bacteria because it goes through the lymphatic system, and then they can be trapped in the lymph nodes. So once they are in the lymph nodes, no, the lymph nodes will react against them. It will create a granuloma around it. So it will try to contain it, no. And then you will notice there are some pink center there's some pink lesions, no, in the center of the granuloma. These are what we call the um, eosinophilic radiating clubs. No, these are pathognomonic for Actinobacillus. So when we say pathonom, pathognomonic, they say yung G silent daw yun, no? So these are pathognomonic, meaning that if you see these lesions, then you can automatically say this is caused by your Actinobacillus pleuropneumoniae. So this is basically the bacteria being contained by the neutrophils and other inflammatory cells, your macrophages, etc., forming the granuloma around the bacteria. For the diagnosis, treatment, and control, you have the exudates. You can gram stain them and then you can grow them on blood agar. Early treatment is best. So you can provide these antibiotics that can kill the gram-negative bacteria. Avoid harsh dry feed. This is in ruminants. Good fouling hygiene naval disinfection in horses, no, when they are born. And then in swine, routinely we can perform ELISA to check the serology and elimination of infected breeding animals, and provide mass medication in exposed herds. That is the common practice, pero 'di ba, we are discouraging the abuse of antibiotics. Okay, so far that's it for Actinobacillus. Any questions with regards to this?